My name is Alex Urban, and I will be performing a presentation about the different nutrition levels of ammonium nitrate and sodium phosphate on Arthrospira platensis. According to the EPA, as climate change becomes more prevalent, severe weather will increase, resulting in more rain and storms, which cause more pollution and stormwater runoff. Within the stormwater, it contains fertilizers, pesticides, as well as other chemicals. Within these, they contain nitrogen and phosphorus, which allow for great plant growth. Through stormwater, they travel to all bodies of water and eventually end up in the ocean. The algae present in the water reacts to the nitrogen and phosphorus like other plants, allowing it to grow. The algae grows and reproduces. When the algae die, they decompose to the bottom of the pond, where algae gets eaten up by bacteria. A study conducted by the Department of Ecology and Evolutionary Biology in 2012 tested algae types with different nutrient levels. The algae that was enriched was eaten the most. This is bad because when bacteria eats algae, they are using up the oxygen in the water and creating dead zones. Dead zones are locations where there is no oxygen at all. With a lack of oxygen in the water, the marine life that lives there is without it and tend to die. This is why algal blooms are bad, and stormwater runoff needs to be dealt with. A last study that was conducted by the Freshwater Biological Laboratory in 1996 tested limited nutrient levels of algae, which resulted in algal growth being depleted or having none at all, resulting in no bacteria and no dead zones. All of this led me to my experiment. The purpose of my experiment is to test the varying nutrient levels of algal growth, to later be explored in other projects on how to create a solution to decrease the algal blooms. My research gap is that the algae Arthrospira platensis has never been tested for the growth of varying levels of ammonium nitrate and sodium phosphate. This introduces my question of what levels of ammonium nitrate and sodium phosphate concentrations allow for the best growth of Arthrospira platensis. My hypothesis is that the water containing the highest levels of nitrogen and phosphorus levels will produce the best growth possibilities. To begin my procedures, I will be adding 500 milliliters of distilled water to 500 milliliter Erlenmeyer flask, and then I will be adding different concentrations of ammonium nitrate and sodium phosphate to the correct Erlenmeyer flask. Later, I will be adding 8 grams of sodium bicarbonate to increase the pH level for the best growth of spirulina. Then we'll add 0.5 grams of salt to the container. Lastly, I will add the 8 milliliters of spirulina to each Erlenmeyer flask. Then I will stream a tube from an air pump into the Erlenmeyer flask to allow for air to get into the algae and allow them to grow the best. To record my data, I will use a spectrophotometer set at 670 nanometers. I will set a blank using distilled water, and then I will add 50 microliters of one sample to a cuvette and record the information for each sample. I will do this all 5 times for all samples. I will repeat this procedures 2 times a week for the next 2 weeks in order to have 4 times of information gathered. This is an example of a graph that I would use, showing all 4 testing times and all 5 concentrations. As the trials went on, you can see how the light passing through decreased as algal growth increased. As of now, I have not done a proper test. Because of trial and error, my first experiment did not succeed, neither did my second one, and I had to dispose of the materials. Secondly, I had to revise my experiment by rewriting my experiment and changing the materials. Now I'm just waiting for my algae to arrive so I can try for a 3rd time and see if I can get my actual data. By the end of January, I will have finished all my trials, having done 3 trials in a total of 6 weeks. I will be finishing near the middle of January, giving me enough time to evaluate and graph all of my data. And these are my sources. Thank you for listening. Have a good day.